So, today we'll talk about disorders of sexual development. Before we start this topic, we'll just see the normal anatomy of development of this sexual organs. So, by 4 to 5 weeks, we are not sure what organs are going to develop in the baby. So, by 4 to 5 weeks, after which with the stimulation of the Sertoli cells, there will be production of AMH if there is a presence of SRY gene. Presence of SRY gene leads to differentiation of male sex organs, and absence of SRY gene is going to create female sex organs. So this is the theory which have been identified till now. So everyone was thinking if there is SRY gene, then it will be male. If there is no SRY gene, then the baby is going to be female. But now there are many other genes which are responsible, which I'll be discussing later. Now, coming to the normal sexual development. So, how does one identify themselves as male or female? So, if I am identifying myself as a female, that is known as gender identity. So, how does this gender identity comes from? So, basically, there has to be some genetic sex which is determined by the chromosomes present in the body, either XX or XY. This will be known as genetic sex, which is the chromosomal sex. So, this presence of XX or XY is going to, lead to the development of the gonads, either the testes or the ovaries, which determines the gonadal sex. If there is a testis, which is the gonadal sex, male. If there are ovaries, then the gonadal sex is female. Okay, and this gonadal sex, testes and ovaries are going to produce certain hormones in the body, and these hormones lead to the development of the external genitalia, internal genitalia, and secondary sexual characters. So, all these external genitalia, internal genitalia, and secondary sexual characters will help in assigning one of the gender to the baby. So, for example, if I consider XX, the gonadal sex as XX, which is corresponding to the female, which leads to production of ovaries, and these ovaries are going to produce certain hormones like estrogen and progesterone. They, depending upon the estrogen and progesterone, there will be development of the secondary sexual characters like breast development or pubic hair development, all these which leads to formation of a phenotypic sex like a male or a female. Seeing this phenotypic sex, we assign a gender to that baby as male or female. So that is when the baby identifies themselves as male or female, that is known as gender identity. So, coming to the genetics of this sex differentiation, as I told, till now it was considered that SRY gene is the main gene which helps in identifying whether it's a male baby or female baby. But now we have certain other genes as well. So, this WNT4 and RSPO gene is going to stimulate the granulosa cell, and this SOX9 and FGF9 genes are going to stimulate SRY gene. So, the presence of these two genes is going to give us a differentiation whether it's a male or a female. Now it's not just SRY gene, we have many other genes as well. So if there is SRY gene, that will act on the Leydig cells and Sertoli cells. This Sertoli cells produces the anti-Mullerian hormone, because of which there will be regression of Mullerian duct. The Leydig cells produces testosterone. This testosterone is responsible for male internal genitalia, and this also gets converted to dihydrotestosterone with the help of 5-alpha reductase, and that leads to development of male external genitalia. So, for the development of the external genitalia, we need dihydrotestosterone. Okay, if there is no dihydrotestosterone, then there won't be male external genitalia, that will become female external genitalia. So the development of the dihydrotestosterone is mainly dependent on your testosterone hormone, and this testosterone is produced by your Leydig cells, which is acted upon by SRY gene. That is why the SRY gene is considered to be a very important gene in the development of the genitals. So then now if there is no SRY gene, there will be increased action of this WNT4 genes. This is Wilms tumor 4 gene and R spondylin 1 gene, which leads to development of this granulosa cell, and this granulosa cell leads to the development of the female internal and external genitalia. So this is the normal sexual differentiation. We have to read now the disorders of this normal sexual differentiation. Thank you.